we had a question of splicing and exons and introns and reading frames, and maybe I should demonstrate that by looking at the exon table of Ensemble. So I've opened here a gene page and I selected the golden transcript or the main select transcript, and then go for the exons page. And what opens from there is this. So we have the summary of exons and introns. Here we have in blue, we have the translated sequence. So there's a little bit of a 5' untranslated region here in red. And then we have the coding sequence starting by ATG, coding for the starting methionine. And then there's a longer stretch of 3' untranslated region. And we have the exon sequences incomplete, but then the intron sequences, which are quite a bit longer, they are of the order of several kilobases for each. They have been abbreviated so that we have just 21st and 20 last nucleotides. And one thing in the splicing machinery, which also helps in predicting splice sites. So we need to have the GT and AG dinucleotides in the ends of the intron. They are recognized by the splicing system, and that determines where the cut is happening when the intron is removed. And in this case, it seems we have phases that are changing. The phase of the splice site refers to the fact whether we have the cut happening exactly between the codons or in the first or the second base position. So phase 0 means that we have the cuts happening in the precise in the precise codon boundary, and phase 1 is having 1 nucleotide coming from the left and 2 from the right, and phase 2 is with 2 nucleotides from the left. And here we have phase 0 and phase 1, and in many genes what we see is all the exons being spliced with the same phase. And that is something that is known as the Lego block structure, and that is the reason for many of domain families happening preferably together with other domains. Often these domains have a single exon coding for the whole domain, and when we have the exons when we have the exons happening in the same phase, when we have the exon spliced in the same phase, it means that we can take the exon with the cuts in phase 0 and move it into the context of another exon in the same phase. If we are trying to move the exon, so let's say that we would try to remove the exon 3 from this one, it would mean that we would be splicing phase 1 to phase 0 here, and that wouldn't work, but instead making the splice would change the reading frame in the splice site, and starting from the splice site we would have a missense translation, often a nonsense translation coming from everything after the frame shift. So jumping to a different phase means a frame shift for the rest of the sequence. And that might be tolerable if we are just dealing with the last 15 amino acids or so. They might not be critical for the protein structure, and we can live with a stop codon coming in prematurely, maybe deleting the last 7 or so amino acids. But whenever we have a frame shift somewhere which is 50 or 100 amino acids away from the end, so probably the protein will be non-functional, being severely truncated, or the transcript is going to be removed very quickly by the nonsense-mediated decay because there's a long region in the 3' end which is not being translated, and thus it's free of ribosomes and more prone to digestion. But I may be going to too much detail in this. And most of the time when we look at the translations, so if we take the, let's go for the cDNA. If we take the cDNA sequence from here we see the actual translation, which is shown in the real reading frame. And I'm looking for the mRNA. Okay, downloading the sequence. Just want the coding sequence. And there it is. And I'm copying this, and to verify how it looks like, I'll go to not Google Translate, but the explicit translate tool. Oh, come on. Explicit is a toolkit at the Swiss Bioinformatics Institute, and they have a lot of reliable and good quality tools. So I'm copying the cDNA here, and let's have the compact annotation, and let's do the translation both forward and reverse. We need to consider the reverse strand if we are taking a raw DNA sequence. This time this is the actual coding sequence, so it is the correct strand, but if we take a random sequenced region that we know contains the protein, it might be in the other strand. So depending on how the sequencing goes, we might get one strand or another. And now this is fairly easy to see which is the real coding strand. Open reading frames are highlighted in red, meaning continuous stretches that start with an initiating methionine and go all the way to a stop codon. And because this was the coding sequence, so it is an open reading frame from start to finish in frame 1. And then these others are giving us shorter reading frames or no open reading frames at all, and there's one long reading frame on the reverse strand, but still it's less than half of what we needed, and this is pretty clear. You should see here immediately that this is the reading frame you want. And if you know what is the protein or an homologue of the protein you are looking for, you can do an alignment between the protein and both strands of the DNA, and in that way find where in the DNA you can match the actual protein.